Alright guys, so today we're going to go into fatty acid anabolism. It's the opposite of catabolism. So the goal of today is to understand how triglycerides are made. So yesterday we tried to understand how triglycerides are broken down into energy. But now if we have too much energy, how do we store it as fat? So how is triglyceride made? Okay, so here it is. So fatty acid anabolism, or you could refer them to fatty acid biosynthesis, or the scientific word for it, the technical term for it, is called lipogenesis. So lipogenesis is a technical term to convert, to basically build triglyceride. Okay, lipogenesis occurs in the cytoplasm. Okay, not the mitochondria, but in the cytoplasm. Okay, and fatty acid actually built 2 carbon at a time, starting from acetyl-CoA. If you guys remember yesterday, we break down 2 carbon at a time to become acetyl-CoA, right? But now we're going to do vice versa. From acetyl-CoA, we're going to build it from here, and we're going to continue building 2 carbon at a time. And that's how we do a lipogenesis, okay? And there are 2 main enzymes involved: acetyl-CoA carboxylase and fatty acid synthase. Those are 2 main enzymes involved in regards to lipogenesis. So just a reminder, basically catabolism and anabolism are complete opposites, especially in fatty acid. And here's the mechanism of beta oxidation. In regards to fatty acid catabolism, you go through oxidation, followed by hydration, oxidation again, and then followed by lysis, right? On the other hand, it's going to be flipped. Anabolism or lipogenesis is going to be flipped. Instead of cutting through thiolysis, you're going to undergo condensation when you link them together. And then instead of oxidation, you're going to go through reduction. Instead of hydration, you're going to go through dehydration. And finally, instead of oxidation, you're going to go through reduction. Okay? So that's basically the steps right there. Okay? So again, it's the opposite of beta oxidation. So condensation, reduction, dehydration, and then reduction again. All right. So that being said, let's go over the mechanism together. So the first step involved is we're not going to go into condensation yet. This is before condensation. We have to kind of prepare prepare all our samples first. Like when you're cooking, you have to kind of like let them incubate in seasoning first before you start cooking them. The same thing here. We have to start incubating and prepare the whole reaction first. So the first step in preparing the reaction is that there's an enzyme called acetyl-CoA carboxylase. This particular enzyme here, what it will do is that it will convert acetyl-CoA into malonyl-CoA. This is very important. Basically, all it does is adds a carboxylic acid to the acetyl-CoA. That's pretty much all it is. So carboxylase will add a carboxylic acid. So acetyl-CoA carboxylase. So it uses acetyl-CoA and add a carboxylic acid to it. That's the enzyme. And during the meantime, when it's doing that, it utilizes 2 very important cofactors. It utilizes ATP, which is very important, as well as biotin. It uses those cofactors in order to do that. And you guys may heard of biotin before. So biotin is what
Is that coenzyme?
Biotin is a coenzyme, yes. So it uses biotin. You guys probably heard that from Costco or some other, in your shelf of drugs you could buy. So you could buy biotin right there. And biotin plays a very important to create malonyl-CoA. This malonyl-CoA here is very important that you need to create. After you're done creating malonyl-CoA, the next step involved is adding a protein called acyl carrier protein. Acyl carrier protein will replace the coenzyme A, and so it becomes malonyl ACP instead of malonyl CoA. And here, on the other hand, acetyl CoA will become acetyl ACP. So that's the second step that's involved. So this is the 2 main steps that require before it enters lipid anabolism or lipogenesis. Okay? So it's very important you undergo these 2 steps. So again, going back, step 1, uses enzyme acetyl-CoA carboxylase convert acetyl-CoA into malonyl-CoA by adding a carboxylic acid group, utilizing ATP and biotin. Okay, and then the second step is you're going to add in an acyl carrier protein, which replaces the coenzyme A. So therefore, it becomes malonyl ACP, and the other one will become acetyl ACP. Okay, so that's the two most important steps. Now, after you have these two steps involved, now you can undergo the four main steps, starting from condensation. So condensation is the next step. What you're going to do during condensation is that you're going to take acetyl ACP and malonyl ACP, and you're going to link them together, combine them together. And the enzyme that plays a big role combining these together will be fatty acid synthase. Fatty acid synthase, once that happens, one of the byproducts will be carbon dioxide. Okay? So what's going to happen here, I color code it so it's easier for you guys to follow. The carboxylic acid here will basically interact with the ACP of the acetyl. Okay? So therefore, what's going to happen is that the malonyl, which is shown in green here, is going to be linked with acetyl, which is right here. The byproduct is carbon dioxide. Why? Is because this carboxylic acid group here will become carbon dioxide. Okay? So that's the first step, condensation, linking malonyl, which is shown in green, and linking it with acetyl, which is shown in blue here. Okay? You link them together. All right. The second step is reduction. During reduction, you're going to use a cofactor, NADPH. You guys may have seen this before. We use that in our lab, NADPH. So we're going to use this particular cofactor to reduce this linkage here, acetyl ACP, and we turn that into this particular enzyme. So all it does, okay, is turn this ketone into a hydroxyl. So instead of double bond O, it's going to be a single bond OH. All right. So during this reduction step, okay, it converts the ketone here into the hydroxyl. You do not need to know these words here. It's just for reference, just in case. All I need you guys to know in this particular thing is how to draw it, okay, and what enzyme and byproducts are involved. Like what enzyme and cofactors are involved. That's what I need you guys to know, okay? So again, during reduction, step 2 reduction, NADPH is involved, and that will convert this ketone into a hydroxyl, okay? The next step involved is going to be dehydration, step 3. During dehydration, you're going to input water. You're going to dehydrate. That means you remove water, right? So therefore, water is now removed. You see that? So when you dehydrate, what's going to happen? You remove this hydroxyl group, and that hydroxyl group will become water. During this process, a double bond is now linked here. You see the double bond? So now the electron will go to here and becomes a double bond here. So once this hydroxyl is removed and becomes water, it will create a double bond right here between the beta and alpha carbon. All right, and finally, the last step, well, almost the last step. Finally, it's going to be the last step is going to be reduction once again. During this reduction, all it is you're going to use NADPH again, right? And NADPH will reduce this double bond and now becomes a single bond. And as you can see, you're slowly growing this fatty acid chain. You see that? All right, and that was step 4. And that's the last step. Well, the last step is basically this guy here will repeat the whole step again with fatty acid synthase. So this particular guy right here will link with malonyl-CoA. Okay? So whatever product we have here at the end will link with malonyl-CoA, and it repeats the whole step again until more fatty acid chain is built, 2 carbon at a time. Okay? And that's the final step, is repeating step 1 to 4 again. Okay? That makes sense for you guys? All right. Any question? All right. So just to repeat, I'm going to just go over one more time with you guys. So step step 1 and step 2 are before the main 4 steps. These are the main 4 steps: is condensation, reduction, dehydration, and reduction again. That's the 4 main steps for anabolism or lipogenesis. Okay? But before we enter the 4 main steps, there's 2 important steps, which is a preparation step. The first step in preparation is that it requires to convert acetyl-CoA into malonyl-CoA. In order to do that, there's an enzyme called acetyl-CoA carboxylase. Okay? And this guy utilizes 2 cofactors, ATP and biotin. Okay? Afterwards, the second step for preparation will be adding in acetyl carrier protein or ACP. So acetyl carrier protein will replace coenzyme A. So instead of CoA here, it's going to be ACP. You see that? So that's the second step of prepping. Now that you prep everything up, now you can follow the main 4 steps. The first step is going to be condensation, step 1. Condensation requires fatty acid synthase. So fatty acid synthase, what it does, it's going to merge these 2 together, acetyl-CoA, ACP, and malonyl-CoA, ACP. Link them together into this new molecule. The second step, which is the reduction, NADPH will come in and reduces this molecule at the ketone right here, at the beta ketone. And beta ketone here will become a hydroxyl. So that ketone, double bond OH, will become a hydroxyl, which is single bond OH. Or double bond O becomes single bond OH. Okay? Afterwards, you're going to do something called dehydration. So you're going to remove this OH, and that will become water. And so at the end, once you remove this, it's going to be linkage. It's going to be a double bond right here. Okay? In order to get rid of that double bond, you need to undergo another reduction. Oops. Another reduction right here to remove that double bond. And that's the last step. That's step 4. And you can repeat this step over and over and over until you have a pretty decent size fatty acid tail. Right? So all you need to do to repeat it is take this guy and merge it with melanin ACP. So all you need to do is take this guy and merge it with melanin ACP. In order to merge that, you need an enzyme called fatty acid synthase. So here it is. So fatty acid synthase again, merge with melanin-CoA. You usually repeat the step over and over until you grow this fatty acid tail 2 carbon chain at a time. All right. And then once you have your fatty acid tail, you actually can link it to the glycerol. Okay? And this particular linkage, we call it esterification or dehydration. So all we need to do is undergo another dehydration reaction, remove that water, and now those 2 are linked. So that's how you link fatty acid tail to the glycerol. Cool? All right. All right, so that being said, what I want you guys to do now is go ahead and take the next, relax for the next 5 minutes. Go over your notes, and then I'm going to ask you some applicational questions, okay? And then just a reminder, if you have not done so, please fill out your proteins.
Are the application questions supposed to be on the lecture too?
Yep.
Okay.
If you haven't done so, fill out your proteins.
So for the written part of the exam, you're gonna want us to write the structure, not the name.
Yeah. For this one, you don't need to know any of the name.
Oh, really? The structure is the one that's important.
Now you're doing chemistry.
Yeah.
All right, guys. So this is just a review for you guys. So if you guys are fasting, and that means you're low on sugar, that means your glucose is gone. The hormone glucagon is now expressed, right? And just a reminder, glucagon plays a couple functions in regards to phosphorylation. So in this case, it will phosphorylate pyruvate kinase, blocking it from making pyruvate. Therefore, everything is going to be everything upstream of it is now blocked, right? So it has to continue making glucose, and glucose cannot be further broken down. In addition to blocking pyruvate kinase, it also activates lipase. Lipase, the main purpose of lipase, is to break down triglyceride, your fat, right? It will turn that into glycerol and fatty acid tail, right? So glycerol will come in and it'll help create more glucose. So now you get sugar from the fat. In addition to that, to your fatty acid tail, those guys will become energy. Now, very quickly, today we learned something new. There's a new enzyme called acetyl-CoA carboxylase, and the phosphorylation inhibits acetyl-CoA carboxylase. If you're wondering what acetyl-CoA carboxylase does, well, it's important to create this guy, malonyl-CoA, which is a very important molecule to continue growing the fatty acid tail. And because now you're blocking it, inhibiting it from doing that, therefore you cannot make fatty acid anymore. You cannot make triglyceride. Therefore, all you can do is continuously break down triglyceride, which is exactly what we want, right? When we're starving, we need energy in some form. So now, this enzyme is blocked, acetyl-CoA carboxylase blocked, and lipase is activated. So now your body is forced to break down triglyceride, and if triglyceride cannot be reformed back into this whole form again. So therefore, it's forced to create energy. And that's a good thing, right? We want that when we're starving. We want to break down the fat to become energy. All right, so again, glucagon affects 3 enzymes that we learned today. Okay? Pyruvate kinase, lipase, and acetyl-CoA carboxylase. All right. So that was catabolism there. But let's talk about anabolism. So if you have a high sugar level in your blood, which hormones are induced? Insulin, right? Very good. So insulin is induced. Very good. So you guys remember, insulin, the main purpose is to activate glucokinase. Better yet, it transcribe and translate glucokinase, so the level is increased. And glucokinase plays a big role in promoting glycolysis, right? So promoting the breakdown of glucose into pyruvate. Okay? But in addition to that, because it plays a big role in breaking down glucose and turning that to glucose 6-phosphate, well, now it's pushing it due to equilibrium. Now it's also, even though it's going down, there's other factors that go down too as well. So continue going down to make pyruvate. At the same time, because due to equilibrium shift, it will push out to another direction. In this case, it will help create more glycogen. So in other words, if you have a lot of sugar in your body, you want to reduce it. One way to reduce the sugar is to turn that into glycogen, so glycogen synthase, right? So now you store more sugar in your body. In addition to that, in addition to turning glucose 6-phosphate into glycogen during this process of insulin, it can also further go down and it also promote the equilibrium towards glycerol creation. So if it's promoting glycerol creation, it's creating more glycerol head for triglyceride. That being said, it also promotes pyruvate conversion, so therefore more acetyl-CoA could be made, therefore fatty acid tail could be made. So fatty acid and glycerol combined together is triglyceride. So overall, if you have insulin, it promotes glycogen buildup, which is your stored sugar, right? And then promote fatty acid buildup. So now you have a bunch of triglycerides stored in your adipose tissue, right? So that's if you overeat and you have so much sugar in your blood, this is what happened to the excess amount of sugar. It will turn to fat. Crazy, right? And yes, it will turn to fat. So sugar is able to turn fat. That's crazy. All right. So in other words, insulin is basically a reverse of glucagon. Now to further enhance that idea, insulin actually activates a protein called phosphodiesterase. That's a new enzyme I want you guys to know as well. Phosphodiesterase, what it does, the main function of phosphodiesterase is to remove phosphate from your enzyme. So, another word, protein kinase, which is activated by glucagon, protein kinase adds phosphate, right? This guy, insulin, transcribe this guy, and that guy will remove phosphate. So in other words, doing complete opposite 180 for all these three, right? So insulin will promote phosphodiesterase, which will remove or dephosphorate pyruvate kinase, dephosphorylase lipase, and dephosphorylate acetyl-CoA carboxylase, right? So it does the opposite. So that's what happened there. Yes?
For glucagon, it inhibits those two in red, right? But it activates the one in green?
Correct. It only activates lipase. These pyruvate kinase and carboxylase here are inhibited. That's correct. All right. And that's all I have for you guys. And then I wrote down fibric acid here for some reason. Oh, that's right. Fibric acid plays a role in triglyceride. So absorb. I think just kind of, what is it? Prevent triglyceride uptake, I believe. All right. And I think I mentioned that yesterday. All right, and that's all I have. So what I want you guys to do now, go ahead and review over your notes for the next few minutes. I'm going to set up Kahoot, and then we can begin that. Okay? And then at that point,